Heart failure. Imagine how frightening that diagnosis is. Your heart cannot pump enough blood to your body. You are breathless and tired. A small exertion causes chest pain, and your heart may spasm, leading to cardiac arrest. There are over 500,000 new cases each year. This enormous problem has been studied for decades, and over $1 billion research dollars are spent each year. So we must be getting close to solving it, right? Wrong. Since 1980, survival after 5 years has risen from 35% to just 50%. Let that sink in. 40 years of research, billions of dollars, yet only 15% more people survive 5 years. The problem is most research has ignored the immune system, which is key to long-term survival. When the heart is damaged, immune cells try to heal, but our research shows that proteins released from a damaged heart cause them to recognize it as foreign and attack. This is autoimmunity, and the damage caused by immune cells releases more proteins, creating a cycle of autoimmunity that current treatments cannot stop. To break that cycle, we must find the culprit protein out of many proteins released from a damaged heart, and further, we must find what tiny piece of that protein is being recognized as foreign. There are millions of combinations, and we have to find the right one, the proverbial needle in a haystack. To find that culprit protein, I have taken advantage of a process the body uses to recognize something as foreign or self. Specialized cells digest proteins and then display tiny pieces of them on their cell surface for immune cells to recognize and then determine whether to attack or heal. I took a protein mixture made from human hearts and added it to these specialized cells grown in the lab, and then identified those tiny pieces on display. What I found was most of those pieces came from just one protein, cardiac myosin, the muscle protein of the heart. Now, if those tiny pieces are causing the cycle of autoimmunity, then there should be more immune cells that recognize them as foreign in heart failure patients when compared with healthy people. And that is the aim of my present work. I'm taking immune cells from heart failure patients and healthy people and adding those tiny pieces of cardiac myosin to them, and then measuring how many immune cells are recognizing them as foreign. And with this method, we can now identify which patients have or may develop an autoimmune reaction to their heart. And most importantly, we can identify which of those tiny molecules are being recognized as foreign, those needles in the haystack, which is the first step toward a treatment that can prevent autoimmunity, promote recovery, and allow heart failure patients to live longer, healthier lives. Thank you.